Depression is a complex disorder characterized by a variety of symptoms and often goes hand in hand with high anxiety and stress. Although the onset of depression is sometimes triggered by some type of stressor, oftentimes the root of the problem can be traced back to genetics and/or one's early life environment. Research has shown for many years that a family history of depression greatly increases one's chances of developing the disorder themselves. Additional research has shown that brain development and subsequent psychological health can be critically controlled by one's environment, especially during childhood. Researchers have largely agreed that a combination of nature and nurture in the nature versus nurture debate is at play in regards to the development of depression, anxiety, and various coping strategies. For example, children of depressed mothers are at a greater risk for psychological problems, although this predisposition can be mitigated by early exposure to novel environments. Similar findings have also been shown in rodent studies, where early exposure to novel situations can improve future anxiety and stress-related behaviors. Together, these studies highlight how genetics and environment interact to shape neurodevelopment and therefore responses to stress and lifelong emotional health. In rats, it is possible to breed for high versus low responders, BHR versus BLR. High responders readily explore and exhibit more risk-taking behaviors, while low responders show high levels of spontaneous anxiety and depressive-like behavior. In this study, a cross-fostering system was developed where BLR offspring were raised either by their biological mothers, fostered to other BLR mothers, or cross-fostered to BHR mothers. The pup's development was then closely monitored. This study was split into two main parts, a behavioral analysis and a genetic analysis. Only male rats were used in this study. The open field test is seen here in the right half of the image and consists of placing a rat into a box for a set amount of time and recording time spent in the exposed center, total distance traveled, and time spent grooming. The elevated plus maze is seen on the left side of this image and consists of 4 elevated arms in a cross shape with 2 open arms and 2 closed arms. The rats are then placed in the center, and the time spent in the exposed open arms, grooming time, and amount of stress-induced defecation are measured. The novelty-suppressed feeding test is a repeat of the open field test, except the rats are food deprived, and food is placed in the center of the box. The social interaction test involves placing a rat in an enclosure with a novel male or female rat, and measuring visits to the unknown rat, stress-induced defecation, and grooming time. Lastly, the forced swim test involves placing a rat into deep water and recording the amount of time spent immobile in the water. All of these aforementioned tested traits are known markers of either increased or decreased anxiety and stress in the rats. The genetic analysis part of this study observed differences in the genomic expression pathways in the amygdala and hippocampus between cross-fostered groups and BLR controls. This included tissue collection, RNA extraction, genome-wide expression profiling, microarray analysis, and quantitative real-time PCR confirmation. On this slide, figures A, C, and E were all recorded from the open field test. Figure A shows that cross-fostering did not affect the time spent in the exposed center of the field, as the cross-fostered group data was the same as the other 2 BLR controls. Figure C, on the other hand, expressed a trend where both fostered groups had a decreased amount of time spent grooming compared to the BLR control. The BLR-BHR foster group tended to groom less than the BLR-BLR foster group, although this data was not significant enough to have a p-value of less than 0.05. Likewise, Figure E supported that BLR-BHR fostered offspring expressed less anxiety-like symptoms, in that they traveled a significantly farther distance than the BLR control. The other data from the elevated plus maze is quantified in figures B, D, and F. Figure B showed that cross-fostering did not have an effect on the amount of time that rats spent in the open, more exposed arms. Similarly, Figure F showed that cross-fostering did not have a significant effect on the distance traveled in the maze. These two findings are in sharp contrast with Figure D, whose data shows that there is a significant difference in the amount of stress-induced defecations of the BLR-BH foster group compared to the other BLR groups. In all aspects of these tests, the BHR controls expressed the least anxiety-like behaviors, which was as expected. Collectively, this data shows subtly reduced adult anxiety-like behaviors in the cross-fostered group. The data graphed in figures A and B were collected from the social interaction test. Figure A shows that the BLR-BHR fostered offspring made more visits to the interaction box than the other BLR groups, which suggests that the BLR-BHR foster group is showing less anxiety-like symptoms than the other BLR groups. Figure B similarly showed that the BLR-BHR cross-fostered group had fewer stress-induced defecations than the other BLR groups. Figures C and D are results from the novelty-suppressed feeding test and show that the latency to enter the food zone was not affected by cross-fostering, and the only significant difference was between the BHR control and the BLR groups, which was as expected. Figure D quantifies the amount of time spent grooming and shows that both the fostered groups spent less time grooming than the BLR control, indicating less anxious behavior. Lastly, the forced swim test produced the results quantified in Figure E, which indicates that the time spent immobile in the water was not affected by fostering, and the only group that spent more time actively swimming was the BHR control. The K-means partitional clustering seen in Figure C is the examination of 103 expressed genes, each represented by one colored line. These genes are all expressed in the amygdala and are separated into 4 clusters determined by similarity in expression profiles. The clusters in white represent the BLR controls, while the clusters in blue are from the BLR-BHR cross-fostered offspring. Each of the lines track a gene's expression pattern over development from day 7 to 75 post-birth. The y-axis is showing normalized intensity values of gene expression, with higher positioning indicating more expression for that gene. The black line in each section represents the mean expression trend for that cluster. Gene ontology analysis was performed, revealing that Cluster 1, cognition, and Cluster 3, vesicle-mediated transport, were significantly enriched. Pathway analysis further revealed 123 pathways that were significantly altered by the BLR-BHR cross-fostering, the top 10 of which are listed in Table 2, with multiple involving genes related to glutaminergic signaling, adrenergic signaling, and apoptosis. Microarray analysis was used to record developmental gene expression in the cross-fostered rats and the BLR control. The data represented here in Figures A and B are an extension of the results plotted in Figure C from the previous slide. Figure A displays the gene expression in the amygdala and hippocampus of the cross-fostered offspring compared to the control. The only statistically significant deviation that did not revert to the baseline BLR control was in the amygdala-specific genes of the BLR-BHR cross-fostered offspring. This deviation did not occur until after postnatal day 14. Figure A is supported by the data shown in Figure B, which represents the upregulation and downregulation of genes for the BLR-BHR cross-fostered groups in relation to the control. As can be seen here, there is a significant upregulation of genes in the cross-fostered group starting after day 14, which parallels the data in Figure A. Overall, this paper was well prepared and the conducted experiments were very thorough. One critique is that the experiment should be replicated using female rats, a sentiment that is shared by the researchers themselves in the second to last paragraph of the paper. Acknowledging their own limiting factors is another plus about this paper in that it shows retrospection. One other small critique involves a formatting issue where some of the graphs in the figures do not seem to be organized in a logical fashion, which interrupts the flow of the paper. This is especially evident in Figure 1, where the data from the open field and maze tests are not separated by a column or row, and rather just dispersed randomly throughout. This study further emphasized the importance of both nature and nurture in the development of psychological issues, especially anxiety, stress, and depression. These results from rodents are applicable to humans by indicating that maternal style and early life events are capable of predisposing or protecting individuals from later psychological issues. It is also interesting to note that there are not only changes in behavior associated with the early life events, but also epigenetic alterations in the gene expression in certain parts of the brain with lifelong implications. Although parental cross-fostering is not usually a viable option in humans, it is possible to reduce the risk of future psychological issues by exposing children to novel environments and people from an early age. This includes multiple caregivers, daycare and preschool programs, and frequent outings.